Hello, I'm Dr. Jade Forster from the University of Southampton, and I'm going to talk about my work identifying m6A RNA modifications in neuroblastoma cell lines using nanopore sequencing. So firstly, what is epitranscriptomics and why should we study the epitranscriptome? So epitranscriptomics describes the modification of RNA molecules. There are thought to be over 150 different types of RNA modifications. Most of these are actually unexplored. The most common modification of mRNA is N6-methyladenosine, or m6A. M6A is thought to have a role in mRNA splicing, translation, and stability. M6A is actually regulated through a highly dynamic process of readers, writers, and erasers. If you just look to your right, so you can see there's a writer complex with the main catalyst being the m6A, which has the methyl group. These are then the methyl groups then read by the readers, and this can be really location specific, so if it's nuclear or cytoplasmic, and that can also impact the outcome of the mRNA. So it could lead to alternative splicing or nuclear export out of the nucleus, or even mRNA stability, whilst other times it could cause translation or even mRNA decay. There are also erasers which remove the methylation, and these are FTO and ALK BH5. And as I mentioned, this process is highly dynamic. The process is actually also dysregulated in cancer and thought to be the underlying mechanism behind immune evasion and telomere maintenance. Just to give you some background on neuroblastoma. So neuroblastoma is a common childhood cancer with a diagnosis at a young age of 18 months. There's a really heterogeneous clinical prognosis, with around 50% of the patients have spontaneous regression with limited to no therapy intervention, whilst the other half already present at an advanced stage where their chances of survival are less than 40%. And that's with a regime of chemotherapy and radiotherapy, as well as more novel immunotherapy combinations as well. And this comes down to actually a lack of there's somatic alterations. So there are some MYC-N like amplifications, as well as ALK TP3 and TERT mutations, but these are in no way global. So actually finding novel treatments to treat the diverse biology and genetic differences in high-risk neuroblastoma remains a significant challenge. Recent studies suggest there is evidence that m6A modifications are involved in neuroblastoma biology, susceptibility, and prognosis. So in the past, it's actually been quite difficult to study RNA modifications with traditional technology. Quite often we had to convert to cDNA before sequencing, so we actually lose the modifications of RNA. But Oxford Nanopore sequencing offers us a chance to detect modifications on native RNA molecules. So there's no need for any different chemistry or library preparations. So if you see on the diagram, the unmodified base and the modified base going through the nanopore, these give us a different signal in which we can confer an RNA-based modification. So actually, from one experiment of direct RNA sequencing, we can get transcriptomic information such as full-length isoform, characterization and expression, alternative splicing, differential gene expression or isoform expression, and poly-A tail measurements, as well as being able to reanalyze the data for base modifications. At the moment, there are limited tools for a limited number of RNA modifications, but we expect over the years, as the base caller becomes more sensitive, we will be able to deconvolute this into new RNA modifications. So for our study, we used Oxford Nanopore sequencing to study the epitranscriptome of high-risk neuroblastoma with the main aim of m6A RNA detection. So for our protocol, we use the GridION device. We use the R9.4.1 flow cells, the RNA 002 direct RNA sequencing kit. And for data analysis, we use Minnow for base calling, Minimap2 for alignment, Nanopolish event line, and then m6A net for m6A detection. So for the actual protocol, we grew 2 high-risk human neuroblastoma cell lines, LAM1, which has a ALK mutation, and IMAR32, which has a MICN amplification. The cell cultures were freshly extracted by Trisol, and we attempted poly-A enrichment methods, but actually we couldn't recover enough RNA, so we decided to use total RNA instead, around 5 micrograms. This at the flow cell load-in stage was then split across 4 flow cells per cell line, and we sequenced that for 48 hours. So our results. So for LAM1, we got just over 2 million reads, and IMAR32, 2.6 million reads, with N50 of 1.5 and 1.7 kb. The m6A net output, we got 11,371 sites for LAM1 and 9,740 for IMAR32. But actually we need to filter these scores. Anything below 0.9 is actually a low confidence, so we filter those out, which left us with 143 sites for LAM1 and 489 sites for IMAR32. So this is just an example of a m6A modified gene. You can see along the bottom below the gene, there's a bed file with the location of the m6A modifications for this particular gene. Most of them at the 3' end of the mRNA, some are in the UTR region, and we do see exonic as well. And in other gene examples, we actually have found intronic modifications also. So as I mentioned before, there's 143 sites in LAM1, but this actually was across 88 genes because we found a multiple number in some of the genes. IMAR32 was 489, and that was actually across 148 genes. When we looked at shared genes between the 2 cell lines, 55 of the genes were actually m6A modified were shared between the 2 cell lines. And gene ontology analysis identified biologically relevant processes. So in the example that I've shown, that's from IMAR32, where there was more genes, so we actually got more enriched processes. But actually, with the 55 genes, the process that was highly significant was histone H3K36 methylation. So in conclusion, we use direct RNA sequencing on 2 human high-risk neuroblastoma cell lines to identify m6A modifications using m6A net. Gene ontology analysis suggests an enrichment in biological processes, one of which is that we think could be important to neuroblastoma biology is histone H3K36 methylation. We've actually got ongoing analysis using other m6A tools as a comparison, as well as looking at other RNA modifications such as m5C. Further analysis is needed though to understand what the significance of these RNA modifications is to neuroblastoma biology. And I'd just like to acknowledge all the cancer, such as Little Princess Trust and Cancer Research UK charities, as well as my university and Jane Gibson and Dr. Juliet Gray and Thomas Wynne for the samples as well. Thank you.